Today I would like to ask only one question, and this is a question that is usually asked by economists. Is it all about location? And we are in a crisis, and economical questions are very important. And I don't know if economists have a good answer on that. But we can also ask these questions in the context of host-pathogen interactions. And we are particularly interested in addressing this by correlating the localization of intracellular pathogens with induced host gene responses. Studying host pathogen interactions is not only important to develop novel drugs and vaccines, it is also interesting to do that because we can use pathogens to understand cell biology, because pathogens have evolved over millions of years together with us, and therefore the factors that pathogens use and the factors that are hijacked in our bodies can be understood by looking at host pathogen interactions. Currently, often we find the following situations. Researchers are either interested in looking individually at pathogens. Here you can see some bacteria stained fluorescently in a confocal image, or people are interested in looking at individual cells, or other researchers are interested in studying the responses caused by pathogens when they encounter our body, which is the immune response. I believe that we have to look at all of this together, and not only we have to look at this together in single studies, we also have to look at this together in single experiments. And this is what we are interested in developing. We want to develop experiments that can give us integrative information on all these events in space and in real time. The model pathogen we have been using is Shigella flexneri. I would not like to introduce the pathogen anymore because this has been done in an extensive and very nice way this morning by Guy Tranvanieux. I would just like to highlight the first steps of Shigella entry into epithelial cells that are normally non-phagocytotic. Shigella uses the type 3 secretion system to trigger its uptake, or to zipper, or I don't remember Guy's word for it, to trigger its uptake into epithelial cells, injecting these effectors, and then it arrives in an endocytic vacuole. This endocytic vacuole can then be ruptured by the bacterium by a so far unknown mechanism. And this is an absolute key step of Shigella invasion, because only when the bacterium arrives inside the cytosol, it can propagate, it can spread from cell to cell, and it can actually cause disease. So we set out to understand really the mechanism of vacuolar rupture caused by this pathogen. And we were particularly interested in figuring out how fast can this pathogen destroy the vacuole to spread inside the cell and then from cell to cell. And this is a question that is not only of importance for understanding host pathogen interactions, it is also important in cell biology in general, because a lot of research have focused either on vesicular budding or vesicular fusion, but vesicular rupture is basically a mechanism that is not understood. So we believe that using this pathogen, we will also shed light on the cell biological process that is very poorly understood. Vacuolar rupture is used by a number of pathogens. The most prominent character is Listeria. Listeria contains a particular set of proteins that can cause vacuolar rupture. Most importantly, Listeriolysin O, and then some lipases. And it has been found that Listeriolysin O can lead to pore formation after 5 minutes upon uptake, and then it takes 15 to 30 minutes to escape from the vacuole. Recent threat studies from Joe Swanson's lab have indicated that Listeria can actually escape from the vacuoles in about 17 minutes. Other bacteria, such as Rickettsia, also escape from the endocytic vacuole. It takes about 12 minutes after host cellular contact to be internalized. And during this time point, we find already 50% in the cytosol. And Shigella is also rupturing the endocytic vacuole, and electron microscopic studies have indicated that 15 minutes after internalization, there is the bacterium already found inside the cytoplasm. However, I would like to point out that these studies are quite static, and they do not give real-time information. And to get this real-time information, we set up a new assay that was done first initially with Guy Tranvagneux and Caroline Claire, and then later on with Katrina Ray at Imperial College and with Alexandre Bobard in my lab. In this approach, we load cells with a fret substrate. This fret substrate can be cleaved by a protein that is on the surface of the invading bacterium. When the bacterium is entering and inside the vacuole, it is not in contact with the fret substrate, and only when it ruptures the vacuole, it is in contact with the fret substrate that leads to a chemical reaction and then to a color change inside the cell. So how does this basically work? The protein that is on the surface of the bacteria is beta-lactamase or penicillinase, and this can cleave a cephalosporin derivative with a fret donor on one proximity and a fret acceptor on the other proximity. So when the bacterium arrives in the cytoplasm, it can cleave this molecule, leading to a change in the fluorescent signal. We first tested the suitability of this test in the test tube in the fluorimeter by incubating the CCF4 substrate, that's the cephalosporin-derived substrate, either together with purified beta-lactamase or together with Shigella wild-type bacteria that express beta-lactamase on the surface. And as you can see, when you have the substrate alone, you get a fluorescent peak at 525 nanometers. However, when you incubate with the bacteria or with the purified lactamase, you get a shift of the signal to 450 nanometers. We then tested bacteria that did not express beta-lactamase, and we could see that there was actually no shift occurring, indicating that our assay really works. And then we also tested other bacteria that cannot invade epithelial cells, just to show whether our assay is functioning or not. And we found that we always need beta-lactamase on the surface of the bacterium to get a change in the fret signal. The first thing we did then, we tested our assay on epithelial cells. And here we found, when we incubated mutant bacteria that cannot invade epithelial cells, or Salmonella that does not escape from the endocytic vacuole, that about 1 hour after the onset of infection, the cells were all green. We also set up an automated image algorithm together with the group of Spencer Shorty at the Institut Pasteur to quantitate massive amounts of data so that we could investigate for each experiment hundreds and hundreds of cells that could give us statistic information. And you can see in this case, when we have no cleavage of the fret substrate, we have a ratio that is very low, both for the mutant and for the Salmonella. However, when we incubate with the Shigella wild-type strain that expresses the beta-lactamase on the surface, we can see during short time courses of infection there's still a lot of cells green. However, at later time points, more and more cells are turning blue. And we can also quantitate this through our automated image algorithms, which shows that at early time points we have very low ratios of our signal, and then at later time points we have more and more signals at high ratios, indicating the escape from the endocytic vacuole. A strength of our assay is that it can be performed in real time, and this is what you can see in this movie, where you see in trans bacteria invading. You can see in green the fret signal, and then you can see how over time the fret signal change with a defret. These signals can be quantitated, as you can see here. These are just false colors, and then we have just the gray values, and the quantification is even nicer indicated in the rainbow colors. We can then correlate the arrival of a bacterium on a host cell in the trans channel, and we can see how long it takes for the vacuoles to be ruptured by following the lag that precedes the change of the fret signal, indicating that we have a very escape from the endocytic vacuole of Shigella in less than 10 minutes. And this was somewhat unexpected because Shigella does not really contain factors that have been clearly associated with the vacuolar rupture, and this is much faster as Listeria. We then used our assay to play around a little bit and to identify host factors that are arriving at the bacterial entry site before vacuolar rupture, and I'm a little bit sorry that this is a little bit small, but we checked our assay in the context with cells that were transfected with row A, and we could find in the transchannel again the formation of the foci when the bacteria are entering, and then in the red channel we could see the recruitment of this molecule row A to the bacterial entry site. Quantitating this, we found on individual cells the time point when foci were formed, and the time points when row A was recruited to the foci of these cells, of the identical cells, and then we could see at later time points when the vacuole was ruptured by our FRET assay, indicating that row A is tethered to the vacuole before its rupture. And then we looked at other factors, and here we have a factor that is called galectin-3 that was identified by Irit Paz and Philippe Sansonetti's group as a marker for bacteria when they exited the vacuoles. And we wanted to use our assay to validate whether galectin-3 is actually a good marker for vacuolar escape of Shigella. And then again, we looked at individual cells. You can see the FRET change in signals in the green and blue channel. In the trans, unfortunately, you cannot see very well the foci formation. And in the red, you can see the recruitment of galectin-3 to the sites of bacterial entry upon vacuolar rupture. And this was becoming more clear when we looked at this on a single cell level, where we could see on a cell when a foci was being formed in the trans channel. We could see in our FRET assay when we saw a change of signal. And then we could also spot the time point when we got an increase of the galectin-3 staining around the bacteria, indicating that this always only occurred upon a change of the FRET signal, indicating that galectin-3 is indeed a marker for vacuolar escape of intracellular pathogens. And I hope that Frank Lafont will talk a little bit more about the function of galectin-3 later today, but I'm not sure if he will do it or not. We can also play with our assay on the bacterial side, and this is what we have been doing. And in this regard, we have been comparing the paradigm of Salmonella entry and the paradigm of Shigella entry. Salmonella is entering into host cells via trigger mechanism. However, it stays inside vacuoles, which is due to factors that are secreted by Salmonella, particularly CYP-A and PIP-B2. However, as I showed to you, Shigella escapes very rapidly from the endocytic vacuole. And it has been shown by the group of Stéphane Mérès in Marseille and David Holton's group in London how CYP-A and PIP-B2 are actually functioning. It has been identified that these factors are targeting the microtubular network inside host cells via kinesins, and then they lead to a particular CYP formation. In the absence of these factors, Salmonella can escape inside the vacuole. So we were interested in stabilizing the vacuole of Shigella, that we can in the future perform more assays with this. So we were asking if CYP-A and PIP-B2 are actually a protection against fast vacuolar rupture. And to do that, we expressed fluorescently tagged CYP-A inside epithelial cells. Then we performed our assay, our FRET assay, when they were challenged with Shigella, and we performed blind and automated image analysis on our assay, indicating that when cells are transfected with CYP-A, with the fluorescent marker M-cherry, they do not allow a vacuolar rupture that is as efficiently as in the absence of CYP-A. We quantitated this and we could see that the vacuolar rupture was down to 40%. We also performed the assay with PIP-B2, and again, Looking at the results we obtained and then quantitating our results, we found a decrease in vacuolar rupture that was about 31%. Co-expressing CIF and PIP-B2 did not have a cumulative effect. And we have to work now on the precise function, how this really works. We also we not only transfected these factors inside the cells, we also expressed them in the bacteria. We checked that they were secreted, and we found similar results. Using other effectors secreted by Salmonella, we found that they actually did not have any effects, but I do not show you the controls for these experiments here. As I mentioned to you, CIF-A and PIP-B2 are targeting the microtubular network, and therefore we wanted to see whether the intact microtubular network is actually important for the stability of the vacuoles that contain Shigella. To disrupt this process or to perturb this process, we disassembled microtubules by a nocodazole treatment at 4 degrees, and we found over time that the bacteria were not able anymore to escape from the endocytic vacuoles as efficiently. Blind assays and quantification of them show that we got a decrease of vacuolar rupture of about 53%. And here I want to show you just that nocodazole treated cells are still capable to take up Shigella, indicated here. So you can still see the foci formation. We also performed electron microscopic studies to show that the bacteria are still inside. But then when we look at our threat experiments, we do not see a change in the threat signal anymore. So concerning the localization or the location, what have I shown you? I've shown you we have developed a novel assay that shows us on a single cell level in real time when pathogens are able to rupture vacuoles. I've shown you that Shigella does this extremely fast. We could identify factors that are tethered to the site before the rupture and that can be used as markers for vacuolar rupture. And then I've shown you that the factors that are secreted from Salmonella, CIF-A and PIP-B2, have a function as vacuolar rescue factors. And furthermore, I've shown you that the microtubular network is involved in the process. And currently we are performing screens to identify the precise host factors that are targeted by the bacteria in this process. So now I would like to address a question I asked in the beginning: does location really matter? And in this concern, I would like to show you the entry of this bacterium, where we can say the hypothesis that depending on the pathogen localization, a distinct host gene expression response can be expected to be triggered. For example, when we have a pathogen on the surface, we can expect that there's a particular subset of genes induced. When we have a pathogen in a vacuole, another subset is induced, and when the pathogen escapes, yet another subset is induced. However, these events are occurring very rapidly, and current assays to study this, such as GFP or luciferase, introduce an uncertain time lag in the assay, and we are basically measuring something at the time point here in the back, and it does not really allow a correlation of the localization with the induced gene expression patterns. Furthermore, gene chips are also not very accurate in this regard, because these steps are very non-synchronous, and the gene chips only give us an average readout, even though there's now some groups that perform gene chips on single cells, but this is something quite tricky to do. To overcome these problems, we set up a novel assay to follow gene transcription in single cells in real time using molecular beacons. Molecular beacons are small oligonucleotide hairpins with a fluorophore on one proximate extremity and a quencher on the other extremity. Upon binding to an mRNA target, the molecular beacons unfold instantly and they start to fluoresce. What we did, we fused promoters that are involved in the immune response to multiple beacon binding sites, and we generated a construct with this, and we put it into cell lines. And then we were able to set up the following assay. We are putting our constructs with the promoter beacon binding site fusions into our cells. We generated stable cell lines. Then we fed these cells with molecular beacons, either by injection or by transfection. And then we stimulate these cells, and we can follow the transcription of mRNA from the specific promoter in real time with the time resolution between seconds to minutes. This assay works on a population of cells, as you can see here, where we performed FACS experiments where we had either non-transfected and non-stimulated cells in the first row. On the x-axis, you can see you can see the signal of the molecular beacons indicating transcription. On the y-axis, you can see a second reporter, GFP, that only shows up at later time points. When we transfect cells with molecular beacons, we can see that they are not stimulated. However, when we transfect cells with molecular beacons that contain a reporter for the interleukin-8 promoter, and we stimulate them with TNF-alpha, we can see an induction from this promoter 2 hours after the onset of the assay. And I would like to indicate here that this is a logarithmic scale. So we are here in 1 log scale, which is already quite a lot. It means a change between 10 to 100-fold. And at later time points, we can also see that the second reporter is coming up, which is the GFP signal. But we were interested looking at this in real time in single cells, and to do this we performed microscopy, and here we looked at the interleukin-8 transcriptional induction in single cells by adding Shigella to the cells that induces a massive pro-inflammatory response, and the changes in transcription are massive. If we look here at the time course, in about like 4 to 8 minutes, you have already in the nucleus a massive increase of signal indicating a strong induction of transcription of interleukin-8. When we use an interleukin-8 mutant promoter, we do not see any increase in the signal. And then we can also see this in a movie where we are just following a single cell lightening up in the nucleus. Of course, it is interesting here to ask what is actually inducing the transcription of interleukin-8. So to do this, Sudhe, the PhD student in the lab who has done a great job in setting up the assay and in performing all these experiments, looked at a lot of movies from wild-type bacteria and then from some mutant bacteria. And so here she added bacteria at time point 10, so before you do not see any changes in transcription. And then once you had the bacteria, you can see from single cells how interleukin-8 is transcribed. When she uses mutant bacteria that do not contain type 3 secretion needles, and she adds them at time point 10, she does not see any signal increases. And very interestingly, when she looks at Shigella strain that expresses an EPA-C GFP fusion that blocks the secretion of the apparatus, in this case, she also doesn't find any increase of the induction, indicating that type 3 secretion is actually required to allow rapid interleukin-8 transcription in single cell. And blocking the type 3 secretion system inhibits the induction of the pro-inflammatory response at these early steps. We were also interested to correlate then the localization of the bacteria with the induction of the gene response. And to do this, we were combining the assays I presented to you. So we could look at bacteria that were either in vacuoles or that escaped from the vacuoles based on our FRET assay or based on the marker galectin-3, and then we could look when were specific genes induced. Doing this, we performed the following movies where we had the galectin-3 signal coming up at the time point when the bacterium escaped from the vacuole, and we could correlate the galectin-3 signal with the curve of interleukin-8 induction inside single bacteria. And this is becoming even more evident in the movie, where you can see the galectin-3 signal here, and you can see in red the increase of the transcription of interleukin-8, indicating that the escape from the endocytic vacuole did not alter the increase of interleukin-8 transcription. So with all this information I presented to you, we believe now, and that is our working hypothesis, that the active injection of the effectors and that the foci formation is responsible for triggering the increase of interleukinate induction. What we are also doing currently is we are not following only interleukinate, we are following genes of different promoters involved in the immune response. And with our assay, we are able to develop real-time gene expression fingerprints in single cells. And we believe in the future, perhaps in 5 years, we will be able to deduce from the patterns we are able to see with our assay what kind of stimulus was actually brought onto the cell. But this is something we would like to do in the future. And with this, I would like to close by highlighting the two aspects I presented to you. I showed to you a novel assay for vacuolar escape of Shigella that indicated escape in less than 10 minutes. And I've shown you the instant innate immune response is triggered within minutes after the initial pathogen contact. These assays can be scaled up for screening, and they can be combined to unravel the key steps of host pathogen interaction interplay. And with this, we can identify the factors that are really involved in this process, and we can perform functional analysis. So with this, I would like to thank the people who are involved in the study. Concerning the vacuolar rupture assay, this was started together with Guy Tramwanieux, Caroline Claire, and with Katrina Ray at Imperial College. And then it has been continued by Alexandre Bobin. The molecular beacon transcription assay has been developed by myself and by Moussa together with Soudé and with Geneviève Janvier. I would also like to thank the platform Imagerie at the Institut Pasteur, particularly Anne Dankert, who has developed the algorithms for automated analysis. And I would like to thank you for your attention.
Thank you, Jost, very much for this. Any questions? Right.
Thank you, Jost, for this nice talk. I was very interested by the fact that the type-free secretion system is important for IL-8 production. So IL-8 mRNA expression is known to be under the dependence of the NOT signaling pathway that is activated by peptidoglycan released. So does it mean that the peptidoglycan should have to go through the type-free secretion system to activate the NOT signaling and then IL-8 mRNA expression?
Similar things have been shown for Helicobacter. I mean, I don't want to go into the mechanism. We have not studied that yet. That would be one option. The other option that I brought up was at the entry, what we find are these massive foci formation. So there may be, when you have this kind of massive foci formation, there may be the possibility of some little incidents that already peptidoglycan could go into the cells, even though this does not represent a real rupture of the vacuole.